Welcome back, everyone, to another episode of Christianity on the Spectrum. This is a podcast of around, at this point, 12 years of ethnographic research covering around 26,000 autistic Christians and ex-Christians in online subcommunities, and over 600 interviews with autistic Christians and ex-Christians. And on this podcast, we like to talk about autism and Christianity and their intersection, or sometimes just autism and sometimes just Christianity. On this episode specifically, though, we are going to be talking about just autism, and more specifically, autism in animals. So this is a question I've been asked a few times. People have been like, John, can you tell me what you think about this study? Because I think this is great and it's going to change my life. And it's a mouse study. And I have to go, hold on, buddy. This is a mouse study. Let's pump the brakes. Calm down. And then sometimes I post about it on Twitter and people are like, why do you hate mouse studies? I don't hate mouse studies. Anyway, this is a whole thing. And I have not yet fully articulated my thoughts on mouse models. And luckily, I have someone who is way more qualified than me to talk about animal models and things like that. So to discuss that, we have Kelly. Kelly, why don't you introduce yourself?
Hi, everybody. I'm Kelly. I'm a friend of the pod. I am also a test subject of John's because, if I have to use a bunch of lab animals, I might as well repay that somehow by being a lab animal myself. But yeah, I have a PhD in neurodevelopmental biology, and I did my PhD working on a gene that is involved in wiring the fruit fly analog to the spinal cord and also to the brain. That actually is a gene that's associated with autism in humans, and in flies can, at the very least, create neuronal wiring and motor defects. But I have also worked with a wide variety of lab animals. I've worked with frog, I've worked with mice. Like I said, I did my PhD in the Drosophila fruit fly. So yeah, you can say I know a thing or two about a lab animal.
Hell yeah. All right.
Oh yeah,
So
Border.
there have been many autism podcasts that I have listened to about animal models of autism, and I find them deeply frustrating. So hopefully, person who knows about animal models listening to this podcast, you do not find this podcast as frustrating as I find those other ones.
The real thing is,
We're
I
here to
probably
correct.
live in ignorant bliss of this podcast. I'm going to be so for real.
Nice,
I
nice.
don't consume much autism content outside of journal articles from PubMed, so I might be living in ignorant bliss here.
You are living in a blissful ignorance,
That
thankfully.
you're
This
gonna
is
wreck very soon for me.
yes, I am I do not recommend you spend much time listening to autism podcasts. Ironically, saying this on an autism podcast, but personally, I have let's say, a complicated relationship with the autism content sphere. So
The Council of Chalcedon.
yes, my Council of Chalcedon is autism self-advocacy podcasts. Very complicated relationship with them. But yeah, so hopefully we will not recapitulate those same types of errors and do a proper explainer on them. So to start out, whenever someone says mouse autism or autism in mice, that usually brings up some red flags to people, and they go, wait a second, how does a mouse have
Yeah, I'm
autism?
already feeling like,
You know, what are people actually talking about? And I think it's important to clarify, mouse autism is a linguistic shortcut. No one actually thinks mice have autism.
well,
They have
a lot of people do,
okay,
but it's because
okay,
they're not aware of that and they read into
yes.
it too much.
Yes, a lot of people misunderstand what's actually being said. It is primarily a linguistic shortcut to express mouse with traits that proxy autism like traits, or mice with behaviors that proxy autism like behaviors, or mice with genes that produce behaviors or effects that proxy genes, behaviors, and effects in humans. They're
Yeah,
not one to one.
not at all. And also, a lot of those mice, maybe they don't even show all they don't get the entire infinity gauntlet of autism traits. It's only validated in like in each of the models. One of them might only have problems with this one social thing. This one might just have like the learning and memory deficits. This one might have the sensory, but there's not, even amongst all these autism mice, they are not
Yes,
showing all the they just
none of them
they show
met.
some of the traits,
Yeah,
or
none of them
have
met the DSM criteria. No one went through the DSM criteria and was like, okay, mouse, I need you to do the ADOS 2 assessment with me. I'm going to give you a bunch of toys to play with. I'm going to see if you like make faces with them or whatever.
I need to see if these impairments cause significant distress in the home, the family, or the workplace, or two.
Yes. Yes, this is not how you determine it in mice. The way you determine if a mouse has autism or autism like traits is you take the mouse and you put it in a box with a toy train, and then you put it in a different box with some other mice. And if it hangs out with the train more than it hangs out with the other mice, you know that one's got autism. So that's how
And
scientists do it.
that's not actually that different from how, I mean, way oversimplified.
Yeah, but can you actually explain like how
Yeah,
we determine
there.
whether or not these things actually like have these proxy traits,
So,
other than the toy train test, of course.
and it's not always a toy train, but anyway, what John is comedically describing is one type of behavioral test that is used sort of as a proxy for sociability in mice. Which, even saying that, that's weighty. We can get into that later, but they use it as a measure of social activity in mice, and it's called the social versus object, or the object preference test, where they'll put an animal, usually a mouse, in a cage, and then on one wing of the cage there's a novel object, like a block that's like a weird color and a weird shape, some object they've never seen before, and then on the other side a age matched member of their own species. And a lot of these quote unquote autism mice, I feel like I want to develop like a better shorthand for them, because it feels so dishonest calling them that, but what I would actually have to say is so much more verbose. Um,
Exactly. This is
anyway,
why
though,
people say autistic mice, because the actual articulation is like 30 words.
yeah. Anyway, though, for shorthand now, a lot of these mice models of autism, compared to wild type peers, the wild type ones usually are more interested and spend more time around the other mouse. Whereas one of the differences observed in some of these mouse models of autism is that they spend less time around the other mouse and are more interested in the inanimate object. And that is one test in a series of tests that are used to determine if a mouse is a model of autism. Um, mind you, all of like there is not, I don't think there's any mouse model where all of these tests have been performed, but there are tests of social. So yeah, the object preference test is a test of sociability. There are also there are some other tests for social activity. Then there's also tests for different cognitive functions. So there are tests of cognitive rigidity. One of those that's kind of interesting is maybe they'll train the mouse to like press the green button and it'll get food, and then we change the rules of the game how long till they realize like, oh, we got to switch and change what we do to this changing situation. So they have certain tests like that for cognitive rigidity, a variety of learning and memory paradigms. There are also tests to test sensory motor gating, which is basically figuring out how much of a stimuli of some type of sensory input does the animal need to register it and behaviorally respond to it. So a big one is like the startle response test, where they'll do noises of increasing loudness and see at what decibel the mouse or the zebrafish or whatever flinches at it. For the record, I have a very overactive startle response, like insanely active. So there, yeah, there's the sensory motor gating tests. There's also other behavioral observation. Like another characteristic of autism in people is doing repetitive stimming type behavior, and some of these mouse models engage in repetitive behavior like grooming themselves over and over again, or running around a circle again and again in their cage, burying and unburying objects. Yeah, those are the main ones I can think of.
Yeah, yeah. There are a few other fun ones that aren't. Fun is the wrong word. There are a few other interesting ones that they use. So like
Oh, the
the
motor
the
activity
mouse.
ones. I forgot about motor activity.
Yeah,
That's a
the mouse depression test
mouse
is not.
depression test is crazy. Okay, that one's nuts. And
Could you explain it?
your reader, it does not explain depression. It explains something, but not depression. Anyway, so there are a variety of them, but basically, the overarching theme is subject mouse to some unpleasant situation and just time how long it takes for the thing to just give up. So one of the depression tests I've read about a bunch of these different tests, and one of the ones I've seen is the tail hold test, where you pick the mouse up by its tail. The mouse is not going to like this. Usually they try to, wiggle away, fight their way out of your hand, and you just hold them and you see how long it takes them to stop. And if they stop quickly, that it's inferred like, oh, they're giving up, they're depressed. And another one similarly is like the mouse. I don't remember the proper word because like I focus more on like the genes and molecules and neurons type of science. I don't do the behavioral tests that much, so I don't remember the name. So I'll call it the mouse dunk tank test, where they put the mouse in a pool of water and he's going to swim and try to get out, and then the one good thing about mice though is when they stop swimming they just float, so just like till it stops swimming and trying to get out and just like floats. And those
Yep.
are the mice depression tests.
So listeners to this might be thinking, this is absurd. However, there is method to this madness. Their behavioral tests, you might be thinking, okay, that's not autism. That's a
That's not
mouse just
depression.
behaving weird. That's not depression. That's just a mouse behaving weird. However, there are some very notable things that happen. So, for example, when you modify specific gene sequences that are present in autism and also present in mice, when you have those gene sequences normal, their behavior goes to sort of standard mouse behavior. But when you modify them in the way that it is in autistic people, they start behaving in those ways. And so like the gene testing isn't like quite, or behavioral testing isn't quite as galaxy brained as it sounds. Like there is actually good reason why people do this. They're not just scientists poking mice to see what happens.
I
Genes
mean, I've
do
done that.
influence
That literally
behaviors.
was my first lab rotation in my PhD, studying pain in mice. So I had to poke their little feet and see
Yeah,
the
but
behaviors that they did.
like genes do influence behaviors, and genes influence behaviors in mice and things like that. So it is important to say that while they are kind of screwing around with mice and poking them all day and just watching to see what they do, it isn't like an insane thing to be doing. There is actually methodological rigor, and you can do controls and things like that, and you can modify
Definitely.
variables to ways that you can isolate these behaviors. And then you could be like, okay, yeah, this behavior is a fairly decent proxy for this other behavior that you would find in humans. And so there are two major models that are used for autism. You have the mouse model and you have the zebrafish model. So, can you explain why we use those two models and like what is even an autistic zebrafish? Why fish?
Oh, fish are such cool model organisms. But I mean, let's start with the mice because they're a bit more obvious, I guess. I mean, everybody's always thought when you think of a lab animal, you think of a lab mouse. And a big part of that is just they are a mammal like us. So they have a bit more in common. Though, like I said, even going back to fruit flies, things have more in common with us than you would like to think. But anyway, mice are mammals, so they're decent approximations for us in that way. Um, we've had their genome sequenced for a long time and developed technology to, alter their genetic makeup in a variety of ways, which you cannot do in every type of animal. They're genetically perturbable. They're mammals like us, but unlike us, they're also social like us. Not exactly like us, but they are a social animal and can do some more kind of complex cognitive tasks and what have you, which honestly, to an extent, the fish can do too. But they're a mammal, they have social behaviors,
They have lots of genetic similarity.
they are
They
genetically
Yeah.
perturbable.
Another thing, they have short gestation times. Uh,
Yeah, I mean, even though as somebody who worked in flies, I'm like, dang, everything in mouse takes so long. So for a mouse, they're born and they're able to, she's pregnant for, I think about 21 days, if I remember correctly from my days toiling in the mouse mines. Then I think the babies they have to live with their mom and then they get weaned at 21 days and then they live for 2 years. And I forgot how long it takes for them to be reproductively mature, but it's like a couple of months or something. Whereas flies, generational turnaround, 11 days, birth to death. Um, but for mammals, mice move quick, and we love that for them. But zebrafish, okay, I'm super fish pilled. So a lot of the draw, the one minor con is that they're not mammals, but they are vertebrates and still share a ton of genes with us. Their neurodevelopment and overall embryonic development goes eerily similar to ours. But what's cool with them is a variety of things. One, they're easily genetically perturbable as well. Lots of cool technologies have been developed to either mutate different genes and not just across the whole animal, but like in certain cell populations as well to overexpress genes. You
I
can
think we should explain right now. Genetically perturbable means you can better edit the
alter the gene
specific
in some
genes.
way.
Yes. Yeah. Anyway,
Oh yeah.
continue.
But yeah, so the zebrafish, the other cool thing about them is you can study really, really early stages of embryonic development because the fish lay eggs outside of their body. So you can do different things to the embryo because it's outside of the mother. You know what I mean? So there's that. And then the coolest thing, in my humble opinion, is that their fish larvae, like the baby fish, are very see-through. So you can live image things inside them in real time, and there's all these different ways you can label different cell types with, you can do this in mice too, but you can't live image them because they're not see-through. But in zebrafish you can make them express certain fluorescent proteins in certain cell types and then like see what the cells are doing in the fish while the fish is alive.
It's so cool that you
It's
can do that. That
crazy.
that is my favorite fun fact about zebrafish. Is you can
Yeah,
like
the imaging
literally
capabilities
watch
on
their
zebrafish are
neurodevelopment.
insane.
Yeah. And so these the whole reason we use these is because they allow you to do quick iterations of things. So in science, without animal studies, you would have to basically wait a very long time for humans to gestate and to develop. It would be very cost intensive. Probably be pretty unethical as well to do a lot of these tests on humans.
Unless you're based Dr. Jian Kui He.
Yes, unless you are him, uh, then you just start CRISPRing people for the hell of it.
Yeah,
You
for
know,
those that don't know, he was a scientist who was the first guy to CRISPR human babies. Like
yeah,
he CRISPRed their embryos, and they're like two girls walking around the earth now with altered genes, and he did it not with the consent of his university or I think the parents.
and he is also a generational poster.
Yeah,
Like
everybody should look at his Twitter. It's crazy.
just
One of the most
the
entertaining Twitters I've ever seen.
of all the genetic researchers I've ever followed on Twitter or any social media for that matter, none of them post nearly as well as he does.
Oh, he posts with an A. He posts with an A.
Yeah, he
He
does.
posts.
Yeah, he does. But yeah, so it allows you to quickly iterate. Basically, is a really big one, and also just like test things. A whole lot of science is throwing spaghetti at the wall and hoping some of it sticks. And to do that, you need to do it a lot and with lots of different things. And you need to
You sure
just
do.
try stuff. Like a huge amount of research is just attempting things that will ultimately fail.
Yeah,
And so
I will say, as somebody who does that for her job, it can be quite demoralizing at
oh yeah.
times. Like
And
your
so
whole system of reward is just totally fried.
yeah, it's a wild field, but having animal models allows you to just do this and allows you to iterate quickly. And it also allows you to do much more controlled tests. If you want to fully control the environment of a mouse, it's much easier to do that than it is with a human, right? Like ultimately, your mouse lives in your lab
Oh
and
yeah, no,
doesn't.
they live in a room that is temperature controlled with the exact same day night cycle of their lights. They're all eating the same exact food, kept in the same exact conditions.
Yes, and can't do that with humans. And so the amount of interference that you get with humans is just way, way larger. And the amount of potential interference you get is way larger. And so if you have these sort of proxy behaviors that mimic human behaviors and can be like selectively changed and controlled through different gene expressions, and you can also control their environment, and then you can control for other things like that. This is a fruitful ground for doing actual scientific research, especially when you are looking at things like gene expression.
I'm
One
not
thing,
biased. I'd have to agree with you there.
yeah, what one thing I should mention though, this field is like, I don't know, 20 years old.
Yeah.
It's not like maybe 25, 30. It's not very.
When I just read a review that was recently published, and it was like 20 years of autistic mouse models.
Yeah,
And I remember like, and I think the first ones started becoming like widely in use around like 2015, 2014, 2015, which is funny because that's when I started reading about him. I was there.
I was
Well,
there, Gandalf.
I was there reading the papers when they were coming out.
Yeah, and so this is still a fairly young field in terms of research, and it is still developing and evolving. When you look at autism research and you look at dollars spent on autism research, animal model testing covers a pretty large portion of it. If you
Yep.
look at if you look at the big donors who are funding autism research, most of their money is going into animal models. And that isn't just because they really hate zebrafish and just want to genocide them. That
I
isn't
love killing zebrafish. It's my
right.
favorite hobby.
I mean, I had a post about that a while ago. That was like, autism advocates, we need more money to research into social services for autistic people. Autism parents, we need research for a cure to autism. Autism researchers, I
I
love
really
killing
love killing
zebrafish.
zebrafish.
Editing John here. Do you know who doesn't like killing zebrafish? That's right, the Against Malaria Foundation, for whom this podcast is raising funds. If you like this podcast, or if you find it valuable, if you find any of my research and work valuable and you would like to financially support it, please consider donating to the Against Malaria Foundation. We have a fundraiser going on. So far, we've raised over $20,000, which is awesome. Thank you for everyone who's donated. It has meant an enormous amount to me. Please continue donating to the Against Malaria Foundation. It genuinely helps motivate me to continue doing this work. Thanks. Back to the podcast. So like this is a very large field of research, but it isn't for no reason. It's because these are, while in their infancy, showing promising results and helping us confirm a lot of things that we suspected, and also potentially developing interventions that can be very helpful. So that's sort of like that's the purpose, right? And so I think it might be
Yeah.
helpful, like we talked about how like different genes and different like gene clusters affect behavior. I think we should like cover a few of the common ones. So a really big one is Shank3. You see that one a lot. So the Shank3 mouse is a mouse with a specific set of genetic mutations that reduce their social interaction and cause repetitive grooming. And also spinal cord loss. I'm not sure how that one factors in, but it's one of the things.
I have some educated guesses, but yeah.
Um, yeah, I won't pronounce how I normally pronounce it on this podcast for the sake of politeness. But there is the CNTNAP2 mouse, more affectionately known by it sounded out by my researcher
Yeah.
friends. I don't know anyone who spells it out, but you can do it in your head. And
Scientists are
that
little
one,
freaks. They love
yeah,
inventing the most insane gene names
yes,
and
the
definitely making dumb puns about it.
yes. So this one causes like social deficits, and it also, interestingly enough, causes hyperactivity. And one other weird thing that it causes is seizures. And so this
Let's
is something that
go. Lots
I
going on
find
here.
super interesting, right? Because you have this gene sequence, causes autism-like symptoms, also causes seizures. And one thing that you will find in autism is that some of us have lots of seizures. There isn't a direct correlation, but it is interesting. Like when you think about neurodevelopment and neurodevelopmental pathways and things like that, how a lot of these things are more connected than you might think
Yeah,
and might
genes
realize.
get recycled, let's just say.
Yeah,
All of
and
them have multiple different purposes, so if it's messed up, chances are it's not just affecting one thing.
yeah, and so like hypermobility and autism is an interesting one that's likely
Oh no.
similar in those ways where autistic people often have hypermobility, and this is probably caused by something developmental that is also affecting the neurodevelopmental side of things, which
Yeah,
is very
basically
cool.
the lore on that one is some of these connective tissue disorders like EDS or just hypermobility in general are caused by mutations in collagen, which forms both our ligaments and our joints and stuff, which would affect mobility, but also creates the extracellular matrix that neurons are crawling across when they're wiring up, and the substrate that they crawl across is very important for them to get to the right place. So it's mutation in this one molecule that is important both in the context of joints and also neurodevelopment. Yeah.
Yeah, very cool. So anyway, these are like the ones you will commonly read about. Like there are a whole bunch of other mouse models though that sort of proxy autism and autism like behaviors. how closely do these things actually proxy human behaviors, though? Like, how close are they, and where do they intersect, and where do they not?
Well, I think one of the biggest things to bring up is species specificity, and I think that comes across a lot in tests of social animals. Like, we can't, for example, like mice, they don't communicate with their eyes as much as we do. So there's not really like a, oh man, that mouse isn't holding eye contact. Um, but they have other ways of communicating. Uh, they sniff each other's butts, kind of like dogs. So they have, the nose to butt sniffing, they have nose to nose sniffing. But smell is a way more important way that they communicate. So when you're looking at social things, you have to look at it, how would they socialize versus how would we socialize? So that's
Yeah,
one of
like
the issues.
mice have like really powerful olfactory senses that humans just don't. And so, what does it mean to be missing eye contact in the mouse world? Is it that you are like not picking up on olfactory cues, or is it different?
There's
Like,
an
these are
interesting olfactory thing. This is the fruit flies for later.
oh, let's get into that now.
Oh, let's get into it. So olfactory cue, because we're talking about social behavior and how it could be mediated by different senses in different animals. And smell or olfactory, that's very important in fruit flies as well. And some of the only behaviors that are not the only behaviors, but some of the best categorized social behaviors in fruit flies relate to mating. Generally, the male fly does like a little courtship dance to a lady fly and will like vibrate its wings in a certain kind of way. They also generally will show aggression to other males or and want to fight over the girl fly with them. But there's this fly that has a mutation in a gene that's associated with autism, and when this gene is mutated, these flies start displaying homosexual behavior. They start doing the courtship dance at the other male flies. Um, and when they look at what this gene is doing, turns out it leads to wiring of neurons in their antenna, which are what they use to sense smell. And due to this difference, they're not able to sense chemical cues that fly is a boy and therefore like a no go.
So, is this why we have disproportionately gay autistic people? This is a joke. This is a joke. This is
This is
a
dead serious, though. It's totally a joke.
joke.
Yeah,
Oh,
guys, we found it. We found the gay gene and we found the gay autism link.
yes, exactly. No, but like, so this is one of my frustration with animal model studies is you'll say something like that and you'll be like, wow, isn't this an interesting thing? And then someone in like a science publication or like a science popularizer will be like,
Okay,
we found the
science
gay gene.
journalists, like, honestly, I don't know. I don't know what needs to happen with them, but there's a problem with them in
Yes,
general.
when the
They want
glorious
those
revolution
eyeballs, they want
comes.
those clicks, and they're gonna word things in the craziest ways.
Yes, yeah, and so yeah, I think that's important to point out. Another thing that I think is important to point out is that how animals behave in the wild and how they behave in labs tend to be quite different. And like there are social behavioral differences in labs versus social behavioral differences in wild environments. And so there are like artificial constructs that go on. Like humans also don't live in lab environments. And so, if you're bringing mice into lab environments and analyzing their behavior in the lab, is that how they would
Also on
behave
like
if they
really,
weren't in a
really
lab?
specific tasks that are going to be like a mouse isn't going to be pressing the green button for cheese uh,
Yeah,
in the wild.
until we start putting out like green cheese buttons in the forest, then that's how we'll get them.
Are we going to make like mouse lintier or something? Like
Yes,
surveil all the
exactly.
mice? If I'm
Yes, Mouse Lintier.
actually, well,
um,
They already kind of have that. I mean, the behavioral essays, that a lot of it, they're just being recorded by cameras the whole time doing their little mousy thing. It's like Mouse Big Brother.
Yeah, yeah, it's wild. Um, so those are some critiques. Now, there have been some pretty cool mouse model successes and just like animal model successes in general. One of the very cool ones is the findings around Fragile X, or what used to be called Rett syndrome. I think it might still be called Rett syndrome. I'm not sure.
I've seen it been called both in all the
Yeah.
papers I've read.
Anyway, Fragile X or Rett syndrome is a really interesting case study. So before 1999, we thought that Rett syndrome or Fragile X were forms of autism. So prior to 1999, this is what we thought. In this current day, if you have a male with Fragile X, 46 to 60% of them will meet the criteria for autism. So the behaviors look very similar. However,
Mm.
Fragile X is caused by a very specific gene. So there are gene mutations that are caused that are that were identified and isolated in 1999. And we went, wow,
Or the
okay,
whole genome was sequenced.
yeah.
Wowie.
Turns out this is not autism. Turns out this is something that happens to look a lot like autism, but is actually a genetic developmental disorder caused by this specific gene and this specific gene path. What we then were able to do is build animal models that replicated Fragile X. More specifically, we were able to build a mouse model and a bird model that have been used a lot.
Oh, I didn't know about the bird model. That's
Yeah,
cool.
yeah.
I
There's a
knew about
bird
the
model.
mouse.
Yeah, it's zebra finch.
Yeah, yeah, yeah,
Uh,
yeah.
yeah.
It's the
Anyway,
classic lab bird.
hell yeah. So zebra finch, zebra fish, zebra finch.
Don't confuse them.
Zebras. Where are our zebra models of autism? If it's in the fish and the finch, it should be in there in the actual like real full deal. Anyway, scientists look into this. But yeah, so anyway, we replicated Fragile X by replicating that specific gene anomaly, and then lo and
Yeah.
behold, the animal models do behave sort of like an animal version of something of a human with Fragile X. After that, after we
Yeah.
identified that, we were able to do lots of drug testing and things like that, and we were actually able to identify like different ways to normalize protein synthesis and synapses in these models, which allowed you to do like pharmacological rescues and things like that of animal models with Fragile X, which is cool as hell.
Super
Like, that is
cool.
awesome.
If only it was that
Unfortunately,
good. I was gonna say if only it was
yeah,
that good for the more pure like autism models, but
yeah. But anyway, it's very interesting because you were able to find this thing in humans, replicate it in an
yeah,
animal, and then when you replicate it in an animal, build pharmacological interventions that could
can
effectively
actually help a
intervene in
human.
animal. And so we've tried to translate these into human studies, and there have not been super glowing successes yet. I will say yet,
Not for
because
Fragile X, really. I thought that one was doing better because, like, I was looking at the stats for the
yeah, so the
autism
results are mixed.
mice, like for all of them, and like only two drugs out of the gajillion they tried show promise in human. But that's kind of a very normal
Yeah,
thing.
exactly. And so they're showing mixed results in humans, and there are there's a lot of future work to be done, and it's probably going to need in humans because we metabolize things differently and slower. You're going to need to play around a lot with dosages and with all sorts of other things like that. You're going to have to take into account other sort of non-lab controlled situations that real humans are in, right? Like
Eating, how you're
you can't, yeah,
sleeping,
exactly. And
work
all
is
those things,
stressful.
all those things are going to affect you. And so you need to, the medication isn't going to immediately work, but it has been helping build or at least find good places to start intervention for Fragile X, which is cool as hell. Importantly, these interventions work in animals, and so like the underlying mechanism that they found with like this drug interaction and this specific genetic anomaly is there is an active mechanism there. Like you just sort of have to keep pushing and figure out
Oh,
what it
finding a
is.
mechanism is so cool. Let me tell
But
you.
but like this would never be possible without animal models, right? Like
No,
this type of intervention would never be found on humans because
and
exact
it's
to
that approach, like the where it goes across the levels. You mutate the gene. Okay, what does that do to the brain morphology? Then what does that do to the behavior?
yeah.
Line them
And
all up. Know what molecules are doing it in what cells and stuff. You can't do that with humans.
exactly. And like you said, like there are billions of different drugs and drug combinations that you try to intervene in these situations, and maybe 2 out of the 10,000 you try will work, but
Hey, if they
that's
work though, for
that's
and if they make some people's life better,
9,998 that we know don't work and 2 that we know might work, which is better than anything we had before. And
that's
this is
science,
how
baby.
science works. And this is the system working as it's intended to. This is
Yeah,
the
but I mean
thing
that's
doing
not
what
going to
it's
make
supposed to do.
everybody.
But like the
But no, I
that's
mean that's the thing.
interesting.
Like to find anything new when you're literally shooting in the dark, it takes a lot of shots. But then you do get a thing that works, and that helps a lot of people.
Yeah,
And
so like
having that
bef-
thing is better than not having that thing.
before we recorded this, you gave a great metaphor, which is, you're sort of in the dark, throwing darts in the dark. And what animal studies allow you to do is say, okay, well, the dartboard is actually sort of in that general direction and kind of this far away from you, right? So
Yeah,
the whole
exactly.
point isn't to be like, yeah, this is how you
The
throw the dart
target's right
exactly
there.
to
No,
hit
it's
it.
like,
Yeah,
this area.
it's
You at
like,
least know that the target will be in this area, and you know that it
yeah,
will not be in these areas. So,
yeah. It's not on
you're
the
making
east
a
wall, it's
more
not on the
informed
west
shot in the
wall.
dark.
Exactly, exactly. It makes it so you're not wasting all your time throwing darts in random directions and hoping something sticks on humans. You throw the darts in random directions on animals that replicate quickly, and then use that to be like, okay,
And also, you
now,
use the animals to figure out the mechanisms of how these things work, so then
yeah,
you know what molecules to even hit in the first place.
exactly. Because speaking as a father, I'm not particularly excited about a scientist going into my child, removing their brain, and then looking through it to figure out mechanisms.
And
However,
good for
you
you. A lot of us would not want to do that because
you can do that to a mouse, and you can do that to mice a lot easier than you can do that to humans. Parents typically object when you try to do that to their children. So these are, to discover the active mechanisms, you sort of need to open things up, right? And
that's
opening
so
things up,
real.
and you can't open up humans. However, you can get a close proxy to humans, and you can throw some more spaghetti at the wall. I will just say, R.I.P. to the lab mice, the true soldiers
I
of
used to like.
humanity.
I used to. They call it when you have to euthanize them. They call it sacrifice. Like, oh, we had to sacrifice the mice today. And I used to like pray in church for the mice I sacrificed. I don't know if they have souls, but I was praying for them.
I think that there is like a mouse heaven for all the mice that participate in genetic research for the
They're
furtherance of.
martyrs.
Yes, I sort of
I literally made
unironically
okay,
believe
wait,
that.
this is crazy lore. I literally made a I mean, I'll show it to you, but the people on the podcast can't see. This insane religious icon of a dead dissected mouse.
That is so cool. You
And
send me a picture and I'll
I'll
put
send you
it
a
in the
picture of it.
notes.
But and it was gonna be part of a larger art piece about using like seeing lab animals as sacrifices and martyrs, and then also looking at their body parts like relics and everything being in like kind of an icon type style with like the gold leafed board and
I
lots of
fear
blood. What?
I fear you are cooking like 8,000 degrees hotter than I was anticipating for this episode, but that's awesome.
Are we surprised though?
No, but I think there is definitely something to that. Like, shout out to the mice and the zebrafish murdered for humanity. Um,
Pray for us.
yes, you guys are doing good out there. Hopefully, the river that bisects heaven or that bisects the kingdom come to earth in
I hope you little
Revelation is
guys get
filled with
all the brine
zebrafish.
shrimp you want to eat
Yeah.
forever.
Yeah. Anyway, other successes after that diversion. Other useful things. We largely suspect that autism has a large genetic component. Mouse
I
studies
think
were
we're
able
even
to
beyond
confirm that.
suspect on that at this point, but
Yeah. Well, like we're beyond suspecting, but for a long time, it was basically, yeah, we suspect this. Mouse models allow you to go beyond suspecting and go like, actually, yeah, these are gene sequences or clusters of genes which increase genetic risk factors for autism.
mm-hmm.
One of the reasons we know that is because of mouse models. They help us confirm things that we already sort of knew and wanted to control, because like I said before, you can sort of turn these genes on or turn these genes off, or like insert them or mutate them, and the mouse behaves differently. And so in the
Can't do
presence
that
of
in a
this
person.
mutation, yeah, you can't do that in a person. But also, like, when you do this on a mouse and its behavior changes, you go, okay, like, this actually is an active gene in autism in a way that you basically can't go down in other ways. So it is useful for just figuring out things that we think we know, even if it can't leave things to interventions. But also, it like it they are generally helpful for finding various interventions and just seeing like, okay, is this actually going to have some potential for translation? The way the way that you think about mouse studies or animal studies in general isn't, this is the key. It is, this is an interesting starting point, right? And so
Yeah,
like
and you have to
the
be
thing I get,
specific about like what kind of result is being captured. Like, not, oh, this drug cures mouse autism. You could be like, oh, this drug in mice with this gene that's an autism risk factor can help them handle some level of auditory sensitivity or something, whatever specific thing, whatever specific cognitive process or other behavioral thing is being tested.
yeah, and like the thing to be quite clear about is, this is, let's find interesting starting points, or this intervention might show promise. That is, whenever you read a mouse study, that is the caveat you need to put in front of it. Don't listen to what the science journalists are writing in the paper. Don't
No,
read the press release.
please. Well,
Right?
speaking
Like,
of science journalists, this is a thing that I like to call like the strawberries cause cancer problem. It's a fictional example, but it's not that different from how they do it in real life. There could be some scientific paper out there that says like, oh, this type of chemical found in strawberries when put on cells at way above normal amount present in strawberries makes them grow more in a dish. And then a science journalist will be like, whoa, strawberries cause cancer, never eat a strawberry again. Like, they remove any kind of
yeah,
context specificity out of it.
yeah, and
um
like the thing is, like, this is how you should interpret things, right? These are demonstrating effects in animal models, which have proxies and parallels to human models. Humans are more complicated than mice, but mice are still complex creatures, right? Like they are not just simple guys with nothing out in with nothing to show for them. They have complex rituals and social features. And I'd also just like to point out science isn't worth doing because it has utility. There is just inherent value to knowing things. So even if the only thing that comes out of a mouse model is you learn how mice work better, that's still useful. And that's still research worth doing. It's a good idea to do these things. It's just you should not over interpret them. But there's also a risk of
No.
under interpreting them, right? So there's over
Yeah.
interpretation where you go like
Oh, like
this is
this
going to
thing
change
showed a little bit of
everything.
promise in a mouse. I should
Yeah.
take this right now. No.
Yeah, there have been more than one person I have spoken to who are like into biohacking and buying research chemicals from China who have
No.
on the reading a mouse study experimented with some things and were like, I didn't feel any effect. And it's like
Are you kidding?
yep,
Wait, that's a thing that
no,
happened.
yep.
Oh
I have
no. Oh
now this isn't
no.
a lot of people. This is two people that I have found in the entirety of my research.
Still two people. That's
Two
rough.
people who were like, wow, interesting mouse study. I'm going to try a research chemical on myself. Anyway, so all of this is to say, overinterpretation bad, but there's another problem, which is underinterpretation, which is something that I see in a lot of autism activist spaces, which is a little weird to me. So
And I,
the
I was
thing,
honestly not even aware that like I said, I live in my little scientist corner, which honestly, there's more crossover than you'd think, because a lot of us, let's just say, experience differential neuronal wiring. I certainly do myself, but in my little science corner, me and all the other autistic science people are hanging around and not really that concerned by the use of mice. So I didn't really know that like there was that strong of like an anti-mouse like current in the discourse. I had no clue.
it's it's profoundly weird, and every time I talk to an autistic research scientist, they're like, "What?
I didn't
People
even know
don't
it was
like
a thing. I'm
mouse?"
gonna be so real.
Yeah, and they're like, "What?" So there are all sorts of criticisms of mouse studies. One of them
Which
is
like I have
like,
plenty myself, but let's see
yeah,
what people have.
yeah, but like one of them is the replication crisis has come from mouse studies, and
There's
a lot of mouse research hasn't replicated the effects that they recorded.
yeah,
Uh,
that okay. There are other
super
things
valid
to
criticism,
criticize. No,
right?
okay, so the replication crisis is a real thing, but the way that we deal with the replication crisis, also remember we're saying these mouse models are relatively new. Behavioral assays or behavioral tests rather, they have so many different ways that they can get confounded and all of that. What we need to do is figure out how to standardize these procedures more to make them more replicable, not like throw the whole thing out. And there actually are moves being made to do that. There's moves. One of the problems that people used to have was, oh, like what this research, like if it's researchers watching the mice during the behavioral test, it's like, oh, well, this guy might count that as a nose bump while the other might not. Okay, we get rid of that problem by switching things to videos that can like automatically record these behaviors according to some type of standard parameter. There are also moves to make some of these tests happen where human interference is minimized to get that confounding variable out of the way. So people like are making moves to make these tests more standardizable so they can be more replicatable. And I think that's what we need to do, not say, oh, because there's issues in these tests, let's never do another test again. No, let's make better tests.
Yeah, and criticism that I've heard from advocates is like we should be spending this money on humans and analyzing autism in humans
We
instead
also
of
do that.
mice. Yeah, it's like we also do that, but also the reason we're doing it on mice is because it produces results, and it produces results much faster than it would produce it in humans. Also,
and can inform us on stuff that we can do in humans, like what
yeah,
drugs may work, and even just under like learning more about how the circuitry in autistic brains are altered, which you can't directly, I guess you can kind of look at it in human, but anyway, can't look at it as deep in
yeah,
humans. Some of these interventions, whether it's pharmacological or even certain therapies that are used, we wouldn't know what to be applying to humans were it not for stuff that we learned in the mouse.
yeah. I would also just like to point out that the way science and science institutions work is it's not like there's a big bucket of money called autism and they just like funnel money from the autism bucket into autism research. That's not how research works. That's not
Is
how
that a thing
funding
that
works.
people think?
Yes. Because they're like, well, why don't we just take that money for doing autism studies in zebrafish and put it towards studying the
Honey,
benefits of
those monies
social
are going to come from like different
services for autistic people?
agencies.
Exactly, exactly. It's not like,
And like public health
yeah,
research is often like a totally different ball game than bench science research.
yeah, and very different sources of funding, very different structures. It's not like there's a big research switch that's like human studies or mouse studies, and you're just waiting for the guy to flip it from one to the other. It's not how any of this works. Um, the criticism of, well, we should be spending this on something else is like, well, I mean, a lot of this funding isn't just for autism, right? It's to look for specific types of things within genes and stuff like that. And it's like, well, it just so happens that autism is a useful thing for us to be looking at to demonstrate these types of principles or to look for these underlying mechanisms. But again, it isn't like people can actually choose where their research money is going and like redirect it to the causes they want to. It's also a little weird to be like, well, the problem with this research is it's not the research I want people to do. It's a bit like, the metaphor that Alyssa uses a lot on the podcast is you go to a recipe website and it's like, here's a recipe for making French toast, and you'll inevitably see a comment on the bottom and it's like, I swapped out the sugar for salt and this tasted terrible. It's like,
Oh
yeah,
my
well,
god.
this is a recipe for French toast, not whatever the hell you wanted it to do. Right? And you shouldn't have made that swap. You don't understand what you're doing here. And I think this is like a very unfair criticism that you should be spending it on something else, where it's like, why should we be spending it on something else?
I
There
mean,
is value
okay,
in
John,
this.
look. I feel like I don't necessarily agree with it, but I do think there's an argument to be made at least.
Oh, there is an argument to be made, and the
Like,
argument
oh,
is
you like
you
mice, it's
shouldn't you
gonna
shouldn't be looking at pharmacological interventions for autism. You should be looking at therapeutic interventions for autism.
sometimes
Is
pharmacological can be therapeutic, but okay.
exactly. But like that is the core of their complaint. It's like the reason we do zebrafish models and the reason we do mouse models is because we're looking for interventions to cure autism.
Oh, that's also not even totally true though.
It's not.
Sometimes we want to learn, okay, why did this cell go different way than this other cell? Or what type of cognitive circuitry in the brain got affected and how. And all of the information that we can get from any of those studies aren't just like, oh, what can we do to like obliterate this condition? It also offers us information on how to make the quality of life for people better. Like figuring out that genes mutated in autism don't just create uh, cognitive defects, but also motor defects. So then, like, knowing this now, we could be like, oh, these kids might need like fine and gross motor skills therapy or something.
Yeah, and I'll also point out.
Or if we know like, oh, this like particular cognitive circuit associated with rigidity is overactivated, how will this affect the way I communicate in a therapeutic setting with an autistic patient or something?
And another thing is like, Okay, how do you think seizure medications that are linked to autism work? Right? That's something that's not curing autism. You're just like stopping seizures, right? How do you propose we build that without using animal models, right? How do you propose we look for underlying mechanisms in all these sorts of like sensory issues for autism, right? Like if you want something that will help you to not get overwhelmed, what sort of intervention do you need? And it's probably going to be some sort of medical intervention. This is not because they're so focused on curing autism. This is just a pharmacological intervention to help autistic people. It isn't that this is like wasted money that's just being thrown into the ether. Like these are meaningful
Yeah, it
projects.
definitely is super helpful in finding treatments for maybe not the quote unquote core symptoms of autism, but definitely a lot of the peripheral symptoms like the
Yeah.
digestive issues, the circadian rhythm disruption, the sensory,
Yeah. And so this is anyway. I think mouse studies are fine.
I also think mouse studies are fine.
Animal studies are fine. They do the job they are supposed to do, which is to find starting points for further research. If you recognize that, you will be able to understand what's going on. However, many people do not, and they overinterpret them or they go, we shouldn't be doing this at all. This doesn't make any sense. We're wasting our time. We're not wasting our time. This is just what science is. Sometimes science is slow.
Okay, this is just because people are science haters. They don't know ball.
Exactly, exactly. You need to stop being science haters. Anyway, this has been Mouse Studies. Um,
This
on
has
that
been
note,
mouse studies.
you can find me Christianity on the Spectrum at gmail.com. You can find me on Twitter at Christianity on. You can find me on Blue Sky at Christianity on.bluesky.social. You can join our Discord, of which Kelly is a member. You can donate to the Against Malaria Foundation to support the pod. Hell yeah, malaria nets. We're at like 18 grand since this podcast has been released. That's awesome. Anything for you to plug, Kelly, before we go?
No, I'm
Awesome.
doing
Okay.
good.
See y'all later.